students, this is Senika. Welcome to my channel PrepBiology. So today in this lecture we are going to discuss Meselson and Stahl experiment. Their experiment proves that the DNA replication is semi-conservative in nature. We have the three models of the DNA replication. First is conservative, second is semi-conservative, third is dispersive model. Conservative model shows that when the parental strand replicates, two strands are formed. One strand is of parental type as same as the parental strand, and one strand is of newly formed. Okay, so this is the conservative model. Semi-conservative model states that the parental strands replicates into the two strands, but one strand of the generation is parental type and one strand is of newly formed. Second DNA strand is of parental type and one strand is of newly formed. So this shows the semi-conservative nature. Okay, and dispersive model shows that the replication takes place, two strands are formed, but both the strands are mixture of. This one single strand of one DNA, and this is the mixture of parental plus newly formed. This is also a mixture of parental plus newly formed. So these are the three models of the DNA replication. But Meselson and Stahl states that DNA replication is semi-conservative in nature. So what did they do? Matthew Meselson and Franklin Stahl performed the experiment in 1958. They work on the bacteria E. coli. Okay, so they work on the bacteria E. coli, that is Escherichia coli. We know that the Watson and Crick model states that the DNA having the double helical structure. So, and DNA is formed by the purines and pyrimidines. Purines having adenine and guanine, pyrimidines having thymine, uracil in the DNA. Okay, so we know that in the structure you all can see the nitrogen is present in excess amount, and this nitrogen is already present in the DNA. Okay, because they are present in the purines and pyrimidines. Which type of nitrogen is already present? N14 type of nitrogen is also already present in the DNA. So Meselson and Stahl took the N15 nitrogen, that is the N15 isotope of the nitrogen, which is heavier than the N14. Okay, so N14 is lighter in nature and N15 is heavier in nature. So Meselson grow the E. coli in the N15 medium, and they grow the E. coli in the N15 medium, which is heavier than the N14. Okay, and they found that the strands of the DNA are of N15 type. Okay, so this is the generation 0. When he transfer these N15 medium bacteria into the N14 medium bacteria, so he found the first cycle of the replication. So in the generation 1, this is the generation 1. In generation 1, he found two strands are formed due to the replication, but both the strands are the hybrid in nature, which is mixture of N14 and N15, which is mixture of N14 and N15. They are hybrid in nature. How they find? That the centrifugation takes place in the density gradient, that is the cesium chloride is used, okay, which is used as a density gradient, and the centrifugation takes place. We have the DNA which is hybrid nature having N14 as well as N15. Now Meselson and Stahl, they grew these bacteria into another N14 medium, and after 40 minutes, he found the second cycle, that is the second cycle of replication takes place. The two strands are converted into the 4 strands of the DNA, and they found that in the centrifugal tube, there are the two types of the DNAs are formed. One is the mixture of the DNA, that is N15 and 14 type, and another, both strands are of N14 type only. So they have 4 types of DNA strands are formed. 2 DNA strands are of N14 type and 2 DNA strands are of hybrid type. So they found that in generation 1 they found the hybrid type of, both strands are hybrid type and 15 and 14, and in generation 2 they found the 4 strands, 2 are hybrid and 2 are lighter in nature, that is contains N14 and 14 isotopes. So by this experiment they proved that one strand in the generation 1 strand is of parental type, 1 strand is of newly type. Okay, so this type of strands are seen in the semi-conservative model of DNA replication. Okay, and one strand of this act as a template strand for the next generation. Okay, so then N14 and 14 types are formed in the generation 2. So what is the conclusion of this experiment? That at the succeeding generations, the DNA extract were found to have lower proportion of N15 and higher proportion of N14. Okay, is it clear to you all? Now they conclude that both the strands separate during the replication and one strand act as a template for the synthesis of the newly strand of DNA having N14. So in next generation, when we replicate these strands, the amount of N14 is higher than the amount of N15. So this proves that DNA replication is semi-conservative in nature. So this is all about Meselson and Stahl experiment. If you like this lecture, like, share and subscribe my channel, that is PrepBiology. Keep studying, keep learning. Thank you so much.